Welcome now to the final part of our review and lecture on lipid metabolism. So for this lecture, we shall be focusing on cholesterol metabolism. We'll discuss first the significance of cholesterol. Then we shall be focusing on cholesterol biosynthesis, the regulation of cholesterol synthesis, as well as pharmacological correlation. And then the final part of our lecture is on the fate of cholesterol. What is cholesterol for? Cholesterol has a very bad reputation because we always associate it with the formation of atherosclerosis. Well, as a matter of fact, it does initiate the formation of atherosclerosis, but it does have physiologic functions. One of them is that it stabilizes the cell membrane, as we have already discussed in the third part of lipid chemistry. Cholesterol is also the precursor for important compounds such as bile salts and acids, as well as steroid hormones. Cholesterol is also the precursor for the production of vitamin D. So where does cholesterol come from? There are two possible sources for cholesterol. It can come from our diet, or our body can produce it. Now, virtually every cell in your body is capable of producing cholesterol, but most of the cholesterol are produced by the liver and small intestine. The precursor is shown here in the diagram. It is acetyl-CoA, which after several steps produces now the ring we call the Cyclopentanophenanthrene ring, which eventually will give rise to cholesterol. Let's briefly review the structure of cholesterol. As shown by the illustration, it is made up of four fused rings. So this ring is what we call the cyclopentanoperhydrophenanthrene ring. Cholesterol on the right side of the screen is shown. It actually contains a hydroxyl group at position number 3. So it is an alcohol, as well as a double bond between carbon number 5 and number 6. So it is an unsaturated alcohol. An 8-membered hydrocarbon chain can be seen at carbon number 17, as well as the presence of methyl groups at carbon number 10 and number 13. So in other words, cholesterol can be described as an unsaturated alcohol that contains the cyclopentanophenanthrene ring. How does our body produce cholesterol? We can divide the events into four major processes. The first is the production of the production of mevalonate from acetyl-CoA, followed by the production of the intermediate precursor known as isoprene from mevalonate. These intermediate precursors, called isoprenes, will eventually condense together to form a linear squalene. It is a linear squalene that will undergo the final phase where it will be closed to form now the 4-ring steroid nucleus. So let's start with the first few events. The production of mevalonate from acetyl-CoA. Acetyl-CoA fuses or condenses with another acetyl-CoA, forming now acetyl-acetyl-CoA. This process takes place in the cytoplasm of the liver, as well as the small intestine, as well as the organs that we are actively involved in cholesterol biosynthesis, such as the gonads and the adrenal glands. Acetyl-acetyl-CoA in turn will condense with another acetyl-CoA catalyzed by the enzyme HMG-CoA, forming now HMG-CoA or beta-hydroxy methylbutyryl-CoA. Now this is the same exact precursor that is produced in the formation of ketone bodies. However, this is again produced in the cytoplasm, whereas the HMG-CoA produced in ketogenesis is produced in the mitochondria. However, there is the possibility that these two intermediates can cross-react with each other. This HMG-CoA will undergo a reduction reaction with incorporation of CoAsh, as catalyzed by the enzyme HMG-CoA reductase. The product is mevalonate. Of all the details that we have mentioned, the step catalyzed by HMG-CoA reductase is the most important step you need to remember, because HMG-CoA reductase is the regulatory enzyme in cholesterol biosynthesis. What happens now to mevalonate? Mevalonate will now give rise to the intermediate precursor referred to as isoprene. In the first few steps, mevalonate is phosphorylated 3 times, forming initially a 5-pyrophosphate mevalonate and eventually a 3-phospho-5-pyrophosphate mevalonate. 3-phospho-5-pyrophosphate mevalonate eventually will be decarboxylated and dephosphorylated to form now delta-3 isopentenyl pyrophosphate. This is already an isoprene, but this particular isoprene will still further undergo an isomerization reaction or a structural rearrangement so that the final isoprene that is produced is called dimethyl allyl pyrophosphate. What happens now to this dimethyl allyl pyrophosphate, or otherwise known as isoprene? Dimethyl allyl pyrophosphate will condense with another dimethyl allyl pyrophosphate, liberating 2 inorganic phosphate, forming now the fused product called geranyl pyrophosphate. In turn, geranyl pyrophosphate will condense with another geranyl pyrophosphate with hydrolysis of inorganic phosphate, forming farnesyl pyrophosphate. In turn, farnesyl pyrophosphate will condense with another farnesyl pyrophosphate, forming now squalene. As you have noticed, the steps in this particular phase of cholesterol metabolism involves condensation of isoprene and the longer derivatives of isoprene, where the final step in this particular phase produces squalene. If you notice the structure of squalene, it is almost like cholesterol, except that it is linear. So the final phase is the cyclization of squalene to form now the Cyclopentanophenanthrene ring. This involves more than 100 steps, but there are steps that we need to absolutely remember, and we don't have to remember every step. The landmark reaction here is the formation of lanosterol, the first that contains the cyclopentanophenanthrene ring. And lanosterol ultimately is one of the immediate precursors for the production of cholesterol. So those are the events that take place in the biosynthesis of cholesterol. The most important step that you need to absolutely remember, however, is located in the first few processes. And this, like what I have said earlier, is the production of mevalonate from HMG-CoA. It's catalyzed by the enzyme HMG-CoA reductase. So HMG-CoA reductase is the regulatory enzyme in cholesterol biosynthesis. As what you have seen previously, it catalyzes one of the first committed steps in the pathway. There are 3 mechanisms for regulating cholesterol through this enzyme. The first is repression of transcription, a mechanism similar to how we operate the lac operon, as we shall be discussing in enzyme regulation. We can also control its degradation These two processes are important for the long-term regulation of cholesterol biosynthesis. However, for the short-term immediate regulation, the enzyme can be modified through covalent modification. Let's start first with a long-term regulation of cholesterol biosynthesis, involving now the degradation of a set of proteins that are embedded on the endoplasmic reticulum. So this is a feedback regulatory system, and the rate of HMG-CoA reductase mRNA synthesis is controlled by a sterol regulatory element binding protein. This is located on the endoplasmic reticulum, and it can be cleaved by protein called S1P and S2P. Let's take a look at the details. So in panel A, we can see the complex protein known as sterol response element binding protein. It contains a DNA binding domain. When cholesterol levels are high, this particular complex is protected from proteolysis. However, as you can see, when cholesterol level drops, cholesterol moves away from this complex, and this protein complex is now prone to proteolysis by proteins known as S1P and S2P, releasing a DNA binding domain. This DNA binding domain proceeds to the nucleus, and it binds to the genes involved in the transcription of HMG-CoA reductase. This now results in the expression of the enzyme HMG-CoA reductase, allowing now for increased biosynthesis of cholesterol. HMG-CoA reductase is actually present in the endoplasmic reticulum, where it catalyzes cholesterol biosynthesis. When the level of cholesterol is already very high, this HMG-CoA reductase is the target for proteolysis. It is eventually degraded with increased amount of cholesterol. So these two are for long-term regulation of cholesterol biosynthesis, as I have already mentioned. Now, what about for short-term regulation? This is via covalent modification. When blood sugar is low, glucagon rises, as shown in this diagram. Glucagon, in turn, stimulates the activation of protein kinase, which in turn phosphorylates the enzyme HMG-CoA reductase, resulting to its inactivation. So in other words, when blood sugar is low, cholesterol biosynthesis is inhibited. However, when blood sugar is high, insulin goes up. This results in the activation of a phosphatase that removes the phosphate group from HMG-CoA reductase, rendering the enzyme active. So in other words, a diet that is rich in carbohydrate promotes cholesterol biosynthesis. So to recap, cholesterol biosynthesis is regulated by controlling the enzyme HMG-CoA reductase, which catalyzes one of the first committed steps in the pathway. So the step catalyzed by HMG-CoA reductase is the primary site for regulating cholesterol biosynthesis. Well, as a matter of fact, drugs like the cholesterol-lowering statin drugs inhibit the enzyme HMG-CoA reductase. Through what mechanism? Let's take a look at the structure of Lovastatin versus mevalonate. If you notice, the Lovastatin resembles mevalonate. They are structural analogs, which means they have the same binding site, and that is the active site. So Lovastatin is an inhibitor of the enzyme HMG-CoA reductase by blocking the active site. It is therefore a competitive inhibitor for the enzyme HMG-CoA reductase. When cholesterol is produced, what are its possible fates now? Cholesterol, when stored, is in the form of cholesterol esters. Cholesterol, when degraded, will assume the bile acid. As a matter of fact, we never utilize cholesterol as a source of energy, but rather its degradatory product is a bile acid. And as shown here, cholesterol can also be used as a precursor for the production of hormones such as testosterone, estradiol, as well as a precursor for the production of vitamin D. To recap, what are the fates of cholesterol? Cholesterol can become part of the cell membrane. It's a minor component, or stored in the form of cholesterol ester. It can be excreted in the form of bile acid, and finally it is a precursor for hormones as well as for vitamin D. Let's take a look at how cholesterol is used in the synthesis of vitamin D. Before we go there, let's take a look at the structure of cholesterol versus that of vitamin D. Cholesterol contains 4 fused rings, the Cyclopentanophenanthrene ring. That ring is no longer present in vitamin D because vitamin D only contains 3 rings. The second ring has been opened up. How does that happen? This is accomplished through simple UV light exposure. So cholesterol in the skin, when exposed to UV light, results to the cleavage of the second ring. This now is what we call cholecalciferol, or vitamin D3. So simple UV light exposure actually converts cholesterol to vitamin D. Now this vitamin D still needs further activation. So it will travel to the liver, where it will be hydroxylated in position number 25, producing what is called 25-hydroxycholecalciferol, or 25-hydroxy vitamin D. The final activation occurs in the liver, where it will be hydroxylated further at position number 1, finally yielding 125-dihydroxycholecalciferol, or 125-dihydroxy vitamin D. As shown in this particular pathway, vitamin D is not essential because we are capable of producing vitamin D through simple UV light exposure. Now, cholesterol is also the precursor for the production of steroid hormones, as illustrated here. These steroid hormones include aldosterone, cortisol, DHEA, androstenedione, testosterone, and estradiol. All those compounds share common metabolites and share a common precursor, which is cholesterol. This is actually taking place in the adrenal cortex. The part that is highlighted by the green line represents the formation of aldosterone. This is taking place in the zona glomerulosa. The part highlighted here is taking place in the zona fasciculata, where the end product is cortisol and cortisone. Whereas this part right here takes place in the zona reticularis of the adrenal gland, where the final product is DHEA and androstenedione. Androstenedione eventually travels into the gonads, where it is converted into testosterone, and in the female, the testosterone is eventually converted into estradiol. So it's actually easy to remember. Just remember the acronym GFR, glomerulosa, fasciculata, reticularis, which corresponds to salt, sugar, sex. Salt for aldosterone, sugar for cortisol, and sex for DHEA and androstenedione. Now take note that 21-hydroxylase is a common enzyme in the production of aldosterone and cortisol. some individuals have defective steroidogenesis. They are suffering from a condition called congenital adrenal hyperplasia. This is an autosomal recessive disorder. Now, the most common enzyme deficiency that accounts for more than 90% of congenital adrenal hyperplasia is deficiency of the enzyme 21-hydroxylase. What will be the effect now of the deficiency of this particular enzyme? Now remember that 21-hydroxylase catalyzes the formation of both aldosterone and cortisol in the adrenal cortex. Since this pathway is blocked, the precursors such as pregnenolone and progesterone are all shunted towards the production of DHEA and androstenedione, which becomes a precursor for testosterone production. So in other words, persons with deficiency of the enzyme 21-hydroxylase have elevated levels of androstenedione, which causes elevation of testosterone. Now, for a female infant, the result of this would be catastrophic because it results in the masculinization of the external genitalia. So what you're looking at here is actually not the penis and not the scrotum, but rather the external genitalia of a female infant that is suffering from congenital adrenal hyperplasia. So the scrotum that you're looking at is not actually the scrotum, but rather it is an hypertrophied labia majora that have fused together covering the vagina. And the glans, what appears to be the glans penis is not actually the glans penis, but rather an enlarged clitoris. So that when this individuals who are born with this condition are often assigned with a male gender, when as a matter of fact they are genetically female. Their internal organs are still the ovaries and the uterus. Now, usually the disorder is only detected on the latter years of the childhood because of repetitive urinary tract infection. So in congenital adrenal hyperplasia, prenatal androgen exposure in female has a masculinizing effect on the development of the external genitalia. So infants are often raised as male, and this would have an effect, of course, on the childhood's brain and behavior. Now, how do we correct this particular deficiency? Well, cortisol is given because cortisol production is diminished. And this is to suppress as well the production of ACTH, to diminish now the amount of metabolites that can be used for the production of androstenedione. At the same time, estrogens may be given to physiologically counteract the masculinizing effect of testosterone. And there could be some surgical correction of the fused labia majora. The storage form of cholesterol is the cholesterol ester. The formation of ester is through the attachment of a fatty acid to the hydroxyl functional group of cholesterol as catalyzed by the enzyme ACAT, or acyl-CoA cholesterol acyl transferase, an endoplasmic reticulum membrane protein. So like what I have said, the fatty acid is linked to the C3 hydroxyl group of cholesterol. Now this ester is what we see as cytosolic lipid droplets in cells. Bile salt, an acid, is the degradation form of cholesterol. Now bile salts and acids are very effective detergents. So these are synthesized in the liver, stored and concentrated in the gallbladder. Now, bile salts and acids are discharged into the gut and aids in the absorption of other lipids such as cholesterol, fatty acids, and other fat-soluble vitamins. Let's take a look at the structure of a a bile acid. Now you can see that it contains a carboxyl functional group. Also, the R1 can either contain a hydroxyl functional group. Now, when glycine and taurine are conjugated to the bile acid, what you have now is the bile salt. The production of bile salt occurs in the liver. In the liver, cholesterol is oxidized to 7-alpha hydroxycholesterol, as catalyzed by the enzyme 7-alpha hydroxylase. This requires molecular oxygen, which eventually forms water, as well as the donation of an electron coming from NADPH. Now, the enzyme that you need to remember here is 7-alpha hydroxylase. 7-hydroxycholesterol eventually will undergo several more steps, not that important to remember, but the final bile acids are canocholic acid as well as cholic acid. Let's take a look at the fate now of a bile salt. So the cholesterol is degraded into bile salt in the liver. The liver synthesizes about 0.2 to 0.6 grams per day and recycles more than 95% of those bile salts. Now, bile salts are eventually stored and concentrated in the gallbladder and released in response to lipids that are being digested. So gallbladder bile acids help in the emulsification of those lipids so that they can be absorbed. Now, those bile are eventually reabsorbed, about 95%, or 12 to 32 grams of bile salts are reabsorbed, returned to the liver for recycling, whereas less than 5% eventually are excreted into the feces. Let's summarize the key concepts that you need to absolutely remember. In the first part of our lecture, we have seen that hydrolysis of VLDL leads to the formation of LDL, which transports cholesterol to peripheral tissues and macrophages. In this lecture, we have seen that HMG-CoA conversion to mevalonate is a rate-limiting step of cholesterol biosynthesis. And how is this particular enzyme controlled? So it is regulated by feedback mechanism. It is degraded when the cholesterol level is very high, and by short-term regulation, it is modified through phosphorylation. Cholesterol have different roles. One of the fate is that it will be a part of the cell membrane. It is stored in the form of cholesterol ester, degraded as bile salt and acid, or it can become a precursor for hormone production as well as vitamin D. Thank you for listening and good luck.